Hello everyone, today we'll be reviewing the major steps of transcription and translation in protein synthesis, as well as the basics of DNA and RNA. Let's start with the cell here. Within the cell, you will find chromosomes, and within the chromosomes, you will find DNA. Within the DNA, you will find genes, and within each gene, you will find nucleotides. We're going to focus in on these nucleotides because they play a crucial role in protein synthesis. Nucleotides are the monomers of nucleic acids. And remember, the nucleic acids are referring to DNA and RNA. Nucleotides' job is to code for specific amino acids. And remember, amino acids are the monomer of proteins. In other words, amino acids are what proteins are made of. You can think of it like this. Nucleotides make the amino acids, and amino acids make the proteins. Nucleotides are made of a phosphate, a sugar, and a nitrogenous base. The nitrogenous bases in DNA are adenine, thymine, guanine, and cytosine. Adenine and thymine pair up, as do guanine and cytosine. These pairs are referred to as a base pair. The nitrogenous bases in RNA are the same with one exception. We have adenine, uracil, guanine, and cytosine. Adenine and uracil pair up, as do guanine and cytosine. So taking a look at the bases in DNA and RNA side by side, we can see that the only difference is DNA has thymine and RNA has uracil. I can't stress how important this is to remember. Thymine is only found in DNA and uracil is only found in RNA. And remember, these nitrogenous bases we're talking about fit in this nucleotide structure. Okay, let's take a look at the structure of a DNA molecule. Here we see the nucleotide that is the monomer of DNA and RNA, meaning it makes up the structure of DNA and RNA. DNA has a double strand that we refer to as a double helix. Both strands run anti-parallel to each other, and this just means that they go in opposite directions. The outside of the strand is made of phosphates, the middle is made of the sugar, and the inside of the double helix is where you can find the nitrogenous base pairs. We have a pair because two nucleotides meet in the middle, so hence a pair. Let's compare the DNA structure to the RNA structure, and I'll point out all of the major differences you need to know. As I mentioned, the DNA structure is made of a double strand we refer to as a double helix, and the RNA is made of a single strand. DNA has the nucleotide thymine, while RNA has the nucleotide uracil. DNA uses the sugar deoxyribose, hence the name deoxyribonucleic acid, which is what DNA stands for. And RNA uses ribose, which is why RNA stands for ribonucleic acid. The function of DNA is to aid in gene expression, replication, and transportation. RNA has many functions, but to basically sum them all up, RNA assists DNA. So, how do DNA and RNA work together? What's the whole purpose of DNA and RNA? DNA and RNA work together in a process called protein synthesis. In other words, they work together to make proteins. Before getting into the steps of protein synthesis, those being transcription and translation, let's go over the different types of RNA you're going to see. You will see three different types of RNA in protein synthesis: rRNA, mRNA, and tRNA. rRNA is ribosomal RNA, and the name gives it a huge hint to its function. rRNA's main function is to create the structure of the ribosome. mRNA is the messenger RNA. Its main job is to transport the DNA information or message from inside the nucleus to the ribosome. tRNA is the transfer RNA. Its main function is to decode the nucleotide sequence and create the appropriate amino acid for the code. Basically, it makes amino acids using the codes on mRNA. As I mentioned, there are two major steps in protein synthesis: transcription and translation. Basically, transcription is where DNA makes mRNA, and translation is where mRNA makes proteins. Let's start at the beginning. DNA is in the nucleus and is unable to leave the nucleus, but it somehow needs to find a way to send its DNA information to the ribosome so it can make proteins. In comes mRNA. mRNA comes along to help by taking a copy of DNA's information and transporting it to the ribosome. Hence why mRNA is the messenger. It's sending the DNA message. Here's our DNA that we find in the nucleus. And here is our mRNA. The major process of transcription begins with the enzyme RNA polymerase. RNA polymerase connects complementary DNA bases to RNA bases. So here is our DNA, and here is our RNA polymerase enzyme. RNA polymerase enzyme is going to do two major things here. One, it will unzip the two strands of DNA, and two, it will code and transfer DNA nucleotide bases to the appropriate RNA nucleotide bases. Here we can see the polymerase enzyme unzipping the DNA. As it unzips, it is also creating the mRNA. So let's take a close look at how mRNA is actually being made here. The RNA polymerase only uses one strand of DNA to code. That strand is referred to as the template strand, and it's also called the antisense strand. So this is our template strand of DNA. Remember, the template strand of DNA has nucleotide bases. Here we have C representing cytosine. RNA polymerase is going to ask itself, what nucleotide base pairs with cytosine in RNA? Guanine pairs with cytosine in RNA. So once polymerase enzyme has figured out the complementary bases, it will make them. So it's going to go ahead and make guanine here. By the way, if you didn't get a chance to write the base pairs down, here they are for reference. It's absolutely mandatory that you remember and memorize these base pairs. Okay, so next on the DNA template strand, we have G representing guanine. Polymerase is going to ask itself, what nucleotide base pairs with guanine in RNA? Cytosine does. So it's going to make a cytosine base. Next, we have thymine. What nucleotide base pairs with thymine in RNA? Adenine. Lastly, we have adenine on the DNA template strand. What nucleotide base pairs with adenine on RNA? Uracil. This is how RNA polymerase enzyme makes mRNA out of DNA. So this is our new mRNA strand, and as you can see here, this is where it comes out of the polymerase enzyme. Once mRNA is made, it transports the DNA message to the ribosomes. And remember, that message is the nucleotide sequence it coded from the DNA template strand. I just want to emphasize the big picture here regarding location. Transcription happens inside of the nucleus of the cell. After mRNA is made, it travels to the ribosomes. This is a ribosome. You can find the ribosome on the endoplasmic reticulum. You can also find the ribosomes freely in the cytoplasm. However, you find protein synthesis more commonly in the ribosomes that are on the endoplasmic reticulum. Okay, so mRNA has clearly moved from the nucleus of the cell to the ribosomes of the cell. Here is where translation begins. Translation happens in the ribosomes of the cell. Codons on mRNA code for amino acids. These amino acids make proteins. Remember, amino acids are the monomers of proteins. Codon refers to 3 bases in a row on the mRNA strand. Each codon codes for a specific amino acid. So let's take a close look at this process. Here is our ribosome. Our ribosome is much like a manufacturing plant because proteins are made here. By the way, I haven't introduced our RNA. This is our RNA. Its main function is to make the structure of the ribosomes. Ribosomes are made of proteins and our RNA, hence the name ribosomal RNA. mRNA starts the translation process by entering into the ribosomes at one end, and out of the other end comes out the finished product, the amino acids. Looking at our mRNA, we can clearly see the codon, which is going to have a big role here now as we take a magnified look at what happens within the ribosome. Okay, so here we are within the ribosome. Remember, our mRNA is going into the ribosome. So here's our mRNA strand inside of the ribosome. The mRNA has nitrogenous bases that are organized into codons. Every 3 bases are equal to 1 codon. And remember, each codon translates to a specific amino acid. This process is all possible because of tRNA, the transfer RNA. Transfer RNA comes in and sits on each codon one by one and translates it into an amino acid. For example, here the tRNA will ask itself, what amino acid does AUG code for? Ah, yes, methionine. So it will pop out a methionine amino acid on the other end. One end of the tRNA reads the codes, and the other end pops out amino acids. So next it reads the code GUC. It's going to ask itself, what does GUC code for? Ah, yes, valine. So it's going to make a valine amino acid. The tRNA will go one by one through each code until it hits a stop codon. This stop codon simulates the tRNA to release the amino acid chain and stop coding. And remember, an amino acid chain is equal to a protein. Okay, so let's take a step back and look at the big picture again. We've seen exactly what happens inside of the ribosome. We know the result is an amino acid chain leaving the ribosome. That will eventually become a protein. And we know it was all created with the help of tRNA and, of course, mRNA. Remember, rRNA basically just makes the structure of the ribosome. That's its function. To summarize, transcription and translation are the steps of protein synthesis, and both DNA and RNA play crucial roles in protein synthesis. Hi everyone, welcome. Today we'll be going over the cell cycle. Before we begin, I want to make sure everyone has a solid understanding of what a chromosome and a chromatid are. Here we have a chromosome. And here we also have a chromosome. You see, chromosomes can come in different shapes and commonly are seen like these two here. This circle in the center of the chromosome is called a centromere. This centromere determines how many chromosomes are present. 1 centromere equals 1 chromosome. This long shape here is called a chromatid. A chromatid is a piece of DNA, a DNA molecule. Technically, it's made of DNA and histone proteins. Here we have 2 chromatids. So on the left, we have 1 chromosome with 1 chromatid. And on the right, we also have 1 chromosome with 2 chromatids. Okay, so now that we can visualize the difference between a chromatid and a chromosome, let's move on to the cell cycle. The cell cycle is divided into 2 major phases, interphase and mitosis. Interphase is further divided into 3 phases. The first one is G1, or also called first gap. The second is S phase, also called synthesis. And the third phase is the G2, or second gap. Following interphase is mitosis. Mitosis is broken down into 4 phases. The first phase being the prophase, the second the metaphase, the third is anaphase, and lastly telophase. So let's go ahead and start off with G1 in the interphase. This is where all cells begin. So this is our cell. Inside we have our nucleus, and inside the nucleus we have our chromatin. Remember, chromatin is a piece of DNA. At this point, this parent cell starts off with 23 chromatin from the mother and 23 chromatin from the father. The mother's genetic material is referred to as maternal, and the father's genetic material is referred to as paternal. The first major thing that happens in G1, or gap 1, is the cell is performing its normal tasks. So it's basically just doing its normal job it's meant to do. The second thing is it begins to replicate organelles. It needs to replicate organelles and everything else within the cell, because eventually this parent cell is going to split into 2 cells. These 2 cells are, by the way, called the daughter cells. And these daughter cells need to be identical to the parent cell. So every organelle within the cell must be duplicated so both cells get equal amounts. One of the major organelles it duplicates is the centrosome. The centrosome has a huge role in DNA replication. It's going to assist moving the chromosomes in almost the entire process. Don't get centrosomes confused with centromere. Centromere, remember, is at the center of each chromosome, and the centrosome is referring to the structure outside of the nucleus that eventually creates microtubules that assist with moving the chromosomes. I normally don't like using tricks to memorize things, but in this case, I feel like these two words are super similar. So I like to think of the word centrosome like this. There is the word some in there. There's the word some in centrosome. So I think somewhere out there. So where is the centrosome? Somewhere out there, okay? And of course, by default, centromere is referring to the center of the chromosome. Thirdly, the cell needs to synthesize or make RNA and enzymes that are needed for DNA replication, because DNA replication is our next step. So G1 tends to be the longest phase, averaging about 6 to 12 hours. After G1, the cell now moves into the synthesis phase, or S phase. Keep in mind, we are still in interphase. Your S phase lasts about 6 to 8 hours. The major thing that happens in synthesis phase is DNA replication. In other words, replication of each of the chromatin. So if we took a close-up here and look at the chromatin activity during this phase, you would see every single chromatin duplicate itself. Remember that at the beginning of the S phase, your cell has 46 total chromatin, 23 maternal and 23 paternal. After DNA replication, which is the end of S phase, you have double the amount of chromatin. You now have 46 maternal and 46 paternal, with a grand total of 92 chromatin. It is really important for you to not only understand what's going on in each phase of the cell cycle, but also to keep track of the number of chromosomes and chromatin in each phase. Keeping track of this number, I think, will help you understand the entire cycle better. So after replicating DNA, the cell enters G2 phase, or gap2. First, the cell continues to grow. Everything within the cell must grow in order to have sufficient amount for both daughter cells. Not just sufficient amount, but also it needs to have enough to give both daughter cells equal size and amount of everything. So for example, the cytoplasm, the mitochondria, the centrosomes, they are all going to continue to grow. Secondly, the centrosomes begin to mature and they make microtubules. Also, the centrioles begin to elongate. And by the way, the centrioles are referring to each of the long tubes that make up the centrosome. So essentially, a centrosome is made of centrioles. Next, the cell finally enters mitosis. The first stage of mitosis is prophase. In prophase, the chromatin condense and coil, and when they do this, they form the shape of an X. So if we again take a really close look at the activity within the nucleus, we'll see the chromatin that are exact copies of each other get nice and cozy together, and they eventually form a shape like this. One side of the X is one sister chromatid, and the other side of the X is the other sister chromatid. Remember, these two sisters are the exact copies of DNA. As a side note, remember, in interphase, you started off with 23 maternal chromatin and 23 paternal chromatin. During DNA replication, they all doubled. Chromatin went from a total of 46 to a total of 92. So this cell that entered mitosis entered with 92 chromatin. Also, there is a difference between chromatin and chromatid. Chromatin is referring to a DNA molecule when it is loose, messy, and the DNA itself is unrecognizable. Chromatid, however, is when this same DNA molecule is more structured. So chromatin makes up chromatids. Okay, so going back to the cell in the prophase, remember the 92 chromatids are now condensed and eventually will take the shape of an X. We still have 92 chromatids. However, they are paired up and now make up 46 paired chromosomes. Remember, we count the number of chromosomes by counting the number of centromeres. So here we have 46 centromeres, so we have 46 chromosomes. In prophase, the nuclear envelope also starts to dissolve. Losing the nucleus will eventually make it easier for the cell to split. Lastly, the centrosomes will begin to migrate to the opposite side of the cell. You can also call this the opposite sides of the poles. After prophase, the cell moves into metaphase. Here, the kinetochore form at the centromere. Kinetochores are a protein structure, and they are found on the centromere, but also on the microtubule spindle. Here's an illustration I found online that shows a bit how the kinetochore works. It holds on to the microtubule and locks it in place. So next, the nuclear envelope in metaphase has completely dissolved, and the centrosomes have now reached the opposite sides of the cell or pole. Lastly, and probably the most important, the chromosomes begin to line up at the metaphase plate. So all 46 chromosomes and 92 chromatids that make up those chromosomes will line up. This is the metaphase plate, by the way. And along with those chromosomes lining up, at the same time, the microtubule spindles will attach to the kinetochores on the centromere of every single chromosome. So basically, the centrosomes have these long arms called microtubules, and they will reach out and connect with each kinetochore at the centrosome of every single chromosome. Each chromosome should have a microtubule attached to it from each of the centrosomes. So basically, there's a microtubule attached to each of the sister chromatids. Once all chromosomes are attached to the microtubules, the cell enters anaphase. Here, the sister chromatids are pulled away from each other at both ends. So 46 chromatids are pulled to one side, while 46 chromatids are pulled to the other side. And we are left with 46 chromosomes on both sides. Also, notice there is a mix of maternal and paternal chromatids on both sides of the cell. It's not just paternal on one side or maternal on the other. Secondly, the cell begins to elongate and accommodate both sets of chromosomes. The cell is now ready to enter telophase. First, a nuclear envelope is formed around both sets of chromosomes. The microtubules release the chromosomes and the organelles begin to move into their proper place. So, for example, the centrosomes have no place in the nucleus. They shouldn't be there. So after releasing the chromosomes, it leaves out of the nucleus and goes somewhere out there. Secondly, and this step is really important, a cleavage furrow pinches the parent cell into two identical daughter cells. This process is called cytokinesis. Cleavage furrow and cytokinesis are both very necessary to learn, necessary to identify and understand both terms. So there's this little pinch here and little pinch there. These areas that are getting pinched are called the cleavage furrow. The cleavage furrow will increasingly pinch and sever the parent cells into two daughter cells. Each daughter cell will end up with 46 chromatids, which is equal to 46 chromosomes. Lastly, in telophase, the chromatids will uncoil and get messy again. In this stage, they're referred to as chromatin again. When telophase is absolutely complete, the cell is now able to make its way into the cell cycle again, or leave and go into G0. So G0 is also known as the resting phase, where the cells just chill out and do their normal jobs and really don't have to worry about replicating themselves. Okay, so we've gone over the entire cell cycle, but there are a few other important points you need to know about the cell division. One is understanding growth arrest and checkpoints. There are checkpoints within the cell cycle. There's one between G1 and S phase, one between G2 and mitosis or prophase, and one more between metaphase and anaphase. Cells must pass these checkpoints in order to move on to the next phase. Passing means they have this check off list, and the cells must have everything properly checked off. If it's unable to check off everything on the list, it will be arrested. So if at any point a cell does not pass the checkpoints, they go into growth arrest. Factors that stimulate growth arrest are crowding around the cell, So this is to prevent a tumor growing. If there is not enough space to grow, then the right thing for the checkpoint to do is say, hey, there is way too much growth around the area, so I really don't think you should be contributing to even more growth where we could become a tumor. So basically it stops the cell from replicating itself. Other factors are oxidative stress on the cell. This includes cells that are just naturally aging and getting a bit too old to do their proper job. Infection, insufficient amount of growth factors or nutrients. Also damaged and incomplete DNA and cells that are not properly attached to their kinetochores. So all of these will alarm the enzymes around the checkpoints and stimulate them to cause an arrest on the cell. Other than the checkpoints and the growth arrest, there are 3 different types of states the cells can be in when not in the cell cycle itself. The first is quiescent state. These cells remain dormant until stimulated. An example would be liver cells. They only divide if there is some kind of viral stress or toxic stress or trauma to the organ, like if someone gets stabbed in the liver. It has the opportunity to grow back because all of the cells are able to replicate themselves if stimulated. Next is the senescent state. These cells permanently do not enter the cell cycle. An example is a cell that has DNA that is too damaged for repair, or naturally aged cells that we all get as we get older. These cells' function begins to deteriorate and the cell eventually becomes slower. So we don't want those cells going through the stress of a cell cycle, nor do we want them to be replicated. Lastly, apoptosis. These cells are pre-programmed to die once they have completed their job. So, for example, the cells that line your digestive tract are constantly dying. They die every 2 to 3 days after completing their work. Then new cells replace them instead of them regenerating. Lastly, there are 3 types of cells you should know. One is stable cells. These cells are not often in the cell cycle. They only go into the cell cycle if stimulated. They spend most of their time in G0, the resting phase. This includes hepatic cells or liver cells, epithelium of the kidney tubules, and lymphocytes. Labile cells are cells that are constantly in the cell cycle and never go into the resting phase or G0, right? These cells proliferate often, meaning they reproduce rapidly. Example of these cells are skin cells, cells in the vaginal wall, and cells that are in the GI tract. Permanent cells go into the cell cycle once and that's it. They are birthed from the cell cycle and that's it. These are amitotic cells, A meaning without, so amitotic meaning they don't undergo mitosis. So we can relate some of these states like quiescent state, apoptosis, and senescent state with these types of cells, right? Stable cells are often in the quiescent state. Labile cells undergo apoptosis, and permanent cells are often in the senescent state. Hello everyone, welcome. Today we'll be reviewing meiosis. So first off, I have this chart here that shows how many chromosomes and chromatids are present in a cell at each stage of meiosis. Make note that these numbers reflect the end of each stage. So if you'd like, you can take a screenshot or take a picture of this before we move on. Let's go ahead and get started. Meiosis is broken down into meiosis 1 and meiosis 2. Much like mitosis, there is a prophase, metaphase, anaphase, and telophase. In meiosis 1, we always put the Roman numeral 1 after each phase. In meiosis 2, we also have prophase, metaphase, anaphase, and telophase. And we put the Roman numeral 2 at the end of these, so we know exactly where we are in meiosis. Notice that between telophase 1 and prophase 2, there is nothing in between, like no interphase. We go straight from telophase 1 to prophase 2. However, we do have interphase at the beginning of meiosis 1. Let's do a quick recap on what happens in interphase before the cell enters meiosis. Here is our cell, and here is our nucleus. Inside the nucleus, we have 23 maternal chromatin and 23 paternal chromatin. Remember, maternal comes from the female and paternal comes from the male. There technically should be a total of 46 chromatin in this illustration. However, I didn't want to make it look too messy, so just imagine there are a total of 46 chromatin inside of the nucleus of a cell. The major thing that happens in interphase is cell division. Specifically, it happens in the S phase or synthesis phase. This means all chromatin are doubled because they all make one copy of themselves. Since there is double the amount of chromatin, we now have 46 maternal and 46 paternal chromatin. Okay, so the big takeaway here is that the cell leaves interphase with 92 total chromatin. The cell is now ready to enter meiosis 1. Specifically, it enters prophase 1. Remember, the cell leaves interphase and moves into prophase with 92 chromatin. Now, a few major things happen here in prophase 1. First off, the chromatin condenses into chromatids with their sister chromatids, and they form this X shape. Next, synapsis occurs. This is when the homologous chromosomes pair up. If you can recall, homologous is referring to one maternal and one paternal chromosome pair. So this is our homologous pair, one maternal and one paternal, and they're going to pair up by overlapping each other. Next, the homologous chromosomes undergo a process called crossing over. This is when the two chromosomes exchange DNA information. If we zoom in, we can see the two chromosomes close up. By the way, this pair is referred to as a tetrad. Each tetrad has 4 sister chromatids that make up 2 chromosomes. Each chromosome has alleles. Alleles are a specific copy of a gene. In other words, each allele has the ability to code for a characteristic trait. In a homologous pair, the same alleles on both the chromosomes are located in the same place, one inherited from each parent. Here, AA and BB are going to code for the same thing. A and B are also going to code for the same thing. Watch closely at AA and BB as they exchange DNA information. Now, A and B are going to exchange DNA information. This is the process called crossing over. This is how they exchange DNA information. Keep in mind that there are 46 homologous chromosome pairs in this nucleus, and all 46 pairs are going to undergo the same process of synapsis and crossing over, and all 46 pairs will look different after this process. For example, let's look at another pair of homologous chromosomes. Here, alleles DD and CC exchange places, but not C and D. Oh, and by the way, remember I mentioned that each allele on the same location of each homologous chromosome codes for the same thing? This is what I mean. Here, these two alleles both code for hair texture. Remember, although we only see 3 pairs of homologous chromosomes undergoing synapsis and crossing over, in reality, all 92 chromatids will be pairing up with their homologous pair and undergoing this process together. Now, I can't stress this enough. It is essential that you understand the purpose of crossing over. Basically, it creates genetic diversity. Every single sperm and egg cell that are produced at the end of meiosis will be different from the parent cell and different from each other. This is why siblings can look very different from each other and from their parents, but not identical. So the big takeaway here is crossing over equals genetic diversity. The last thing that happens here in prophase 1 is the nucleus begins to disappear. The most important events in prophase 1 to remember are synapsis, crossing over, and of course, crossing over means genetic diversity. So leaving prophase 1, the cell moves on to metaphase 1. Here, the only eventful thing that happens is the chromosomes line up on the metaphase plate. Keep in mind that these chromosomes can line up in different random variations, which will result in variations of gametes or haploid cells. They can line up like this, or like this, or like this. Also keep in mind that on the right, 23 full chromosomes, and by full I mean a paired, chromosomes are lined up, and on the left, 23 full or paired chromosomes are also lined up, giving us a grand total of 46 total chromosomes. As I mentioned, this is the only eventful thing that happens in metaphase, and it is the most important thing to remember. Aside from that, the spindle fibers elongate and they attach to the centromere of each chromosome. These yellow dots represent the centrosomes, and those are the spindle fibers coming out of them. Next, the cell moves on to anaphase 1. The most important thing that happens here is the homologous pairs are pulled away from each other, each going to opposite ends of the cell. At this point, keep in mind we have 23 chromosomes pulled to each side. So 23 chromosomes on the right and 23 chromosomes pulled to the left. Again, the only important thing to remember here is that the pairs are splitting and being pulled away from each other. After anaphase 1, the cell moves on to telophase 1. Here, cytokinesis splits the cell into two cells. We started with one parent cell and ended with two genetically different haploid daughter cells. Haploid meaning the cell contains a half of the complete set of chromosomes. So if you need to look at the full set for reference, go back to metaphase 1 and anaphase 1 before cytokinesis happens. So each of these haploid cells contain only one set of chromosomes, each set consisting of paired sister chromatids. At the end of telophase 1, we have 23 chromosomes and 46 chromatids in each cell. These cells are ready to leave telophase 1 and move on to prophase 2. In prophase 2, the nucleus dissolves and the spindle fibers elongate. Pretty much the same stuff that happens in mitosis happens here. These yellow dots again represent the centrosomes, and the lines coming out of them are the spindle fibers. The cell next leaves prophase 2 and enters metaphase 2. Here the chromosomes line up on the metaphase plate. This is pretty much all that is important to remember in metaphase, is that the chromosomes, they line up. After metaphase, the cell moves on to anaphase 2. Here the spindles attach to the centromere of each chromosome and pull the chromatids apart. By the way, do you remember the structure that the spindle fibers specifically attach to? The kinetochores, yes. Specifically, the spindle fibers will attach to the kinetochore on the centromere. So this is the most important thing you need to remember in anaphase 2. At this point in anaphase 2, we have 23 chromatids, which is equal to 23 chromosomes here, 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 and here. Side note, in case you forgot, we count the number of chromosomes by the number of centromeres present, so that's why we have 23. After anaphase 2, the cell moves on to telophase 2. Here, cytokinesis will split these two cells into 4 total cells. So these two will each split and make 4 haploid cells. This is the final step of mitosis, but there are a few key points you need to know here. One is each cell will end up with 23 chromatids equal to 23 chromosomes. Each cell is different from one another and different from the parent cell. And lastly, if meiosis is happening in a female, then the 4 cells being produced will be egg cells because females have eggs. And if meiosis is happening in a male, then the 4 cells will be sperm cells because males have sperm. Oh, and one last thing, these 4 haploid cells are also referred to as gametes because gametes are sex cells. I guess what I'm saying here is that mitosis produces somatic cells and meiosis produces sex cells or gametes. Hello everyone, welcome. Today we'll be reviewing Mendel's law and Punnett squares. Let's go ahead and get started. So first off, there are a few terms you need to be familiar with before we start practicing Punnett squares, like allele. An allele is a type of a gene. Each gene has 2 alleles. For example, if eye color is the gene type, then the color would be the allele. So here, brown eyes is one allele, and blue eyes is the other allele. Another example is if hair texture is the gene type, then curly hair is the allele and straight hair is the other allele. So remember, we have 23 chromosomes in our body. Let's zoom into one of these chromosomes and find the alleles in them. Remember, one chromosome is made of two chromatids. One is maternal, coming from the female, and the other is paternal, coming from the male. So here we will find our alleles. Both of these specific alleles code for one type of gene. That gene type is hair texture. This one codes for curly hair, and this one codes for straight hair. When we have two different alleles, this is called heterozygous. If both of these alleles coded for the same allele, in this case curly hair, then it would be called homozygous. Remember, organisms have two alleles for each gene, one from each parent as seen here in this example. These two alleles can either be dominant or recessive. Dominant is the trait that is expressed, and recessive is the trait that is not expressed. In other words, dominant is manifested and recessive is hidden. A gene is represented by a specific letter. In this case, we represent hair texture with the letter H. One of the alleles will be dominant and the other will be recessive. We represent the dominant allele with a capital H, and we represent the recessive allele with a lowercase h. So here we have a capital H representing the dominant allele or trait, and a lowercase h representing the recessive allele or trait. When we say the alleles are heterozygous, we already know that means the alleles are different. This is what it looks like, a capital letter and a lowercase letter together. And when we see homozygous, we know this means both alleles are the same. This is what homozygous looks like. Either we have two uppercase letters together, or we'll have two lowercase letters together. Let's say we're given the following information. Big H equals straight hair, and little h equals curly hair. When the dominant allele is present, it will mask the other allele. We can only have three possible allele pairs here. Big H, big H, little h, and little h. What outcome will we get with the combination of big H, big H? Will we get straight hair or will we get curly hair? We will get straight hair because the dominant allele is present. In other words, the big H is present. How about big H, little h? Again, we're going to get straight hair because again, the dominant allele is present. The big H is present. How about little h, little h? We will get curly hair because there is no dominant allele present to eclipse the recessive. The only way for a recessive gene to be expressed is if it is in this form, homozygous recessive. Because in this form, there is no dominant present to overshadow it. So the recessive allele can go ahead and shine. It can go ahead and manifest. Okay, so this girl has curly hair. What possible allele pairs could she have? Little h, correct. Only this pair will yield curly hair. How about this girl? She has straight hair. What possible allele pairs could she have? Yes, she can have big H, or she could have big H, little h. Both of these would yield straight hair. By the way, we're going to start referring to these allele pairs as genotypes. Genotypes is the letter combinations, and genotype refers to the genetic makeup. The genotype is not visible, as opposed to the phenotype. Phenotype is referring to the characteristic traits expressed by the genetic makeup. The phenotype is observable. For example, this girl's phenotype is big H, or big H, little h. Her phenotype is straight hair. Okay, so now that we know the basic terminology, let's look at some Punnett squares. We'll first look at a monohybrid Punnett square. We use these to examine only one trait. Parent 1 and 2 go on the sides like this. And remember, we're studying one trait or one gene. So one gene will have two alleles. So for each parent, we place those two alleles here and here. What's inside the boxes are the possible offspring outcomes. So each box represents 25%, and of course, it adds up to 100% as a whole. So let's look at our first practice problem. A homozygous cat without fur mates with a heterozygous cat with fur. Fur is a dominant trait. What is the possibility the offspring will have no fur? And what is the genotype ratio and the phenotype ratio? Okay, here's our Punnett square that we're slowly going to fill in. But first, notice one very important piece of information given. Fur is dominant. So by default, no fur is recessive. First off, let's find the variation of genotypes we can have. We can have big H, big H, little h, and little h. These allele variations or genotypes can provide some information for us. Like, we know because we have two capital H's that this is dominant and it will yield a phenotype with fur. We also know it's homozygous because it has two capital H's. For big H, little h, we know this is also going to give us a phenotype of fur because there's one dominant allele present. We also know this is heterozygous because there's a capital H and a lowercase h. For little h, we know it will give us a phenotype of no fur because we have two lowercase h's here representing no fur. There is no dominant allele here to mask the recessive allele. So the recessive allele here is able to manifest. We also know this is a homozygous because both letters are the same. They're both lowercase. So we're going to use this information to find out what genotype fits in the parents of these cats. The genotype of each parent will fit into these four areas here on the Punnett square. Let's work on parent number 1. Here it says we have a heterozygous cat with fur. We also know this cat parent is dominant because it tells us right here the fur is dominant trait. So we need to find a genotype that is heterozygous and will produce a phenotype with fur. Big H, little h fits that description perfectly. Big H, little h is a heterozygous genotype and its combination yields fur. In other words, it will give us a cat with fur. So we're going to put the genotype right here to represent the cat parent with fur. Next, let's work on the second cat parent. Here it says we have a homozygous cat without fur. So we need to look for a genotype that matches that description. Two genotypes are homozygous, but only one of these is going to yield a phenotype without fur. So we're going to put that genotype into the Punnett square to represent the cat parent without fur. Next, we're going to go ahead and fill in these boxes. So big H and little h gives us big H, little h. Little h and little h is going to give us little h, and so on. So remember here that a capital letter represents the dominant trait. And if there is a dominant trait present, it will mask the other allele. In other words, the dominant trait will be expressed. So here we can say 50% of the offspring will have fur because they have a big H present. And 50% will not have fur because there's no big H present. So that answers question number 1. Question 2, what is the genotype ratio? Genotype is referring to the different letter combinations we see. There are only 2 letter combinations here, big H, and little h. So the ratio is going to be 2 to 2, or we can say 1 to 1. Next, the question is asking for the phenotype ratio. Phenotype is referring to the characteristic traits that are visible, like does the cat have fur or does it have no fur? These two with the dominant alleles present will give fur, and these two with no dominant alleles present will give us no fur kittens. So the ratio here is 1 to 1. Just want to add that the genotype and phenotype ratio happen to be the same here, but keep in mind that they won't always be the same. So these are our answers here. Let's try another problem. This problem is from the official ATI TEAS study guide. I included a couple of these so that you can get a sense of what they're looking for and how they word their questions. Use a Punnett square to show the likely phenotypes of crossing two heterozygous tall pea plants. The allele for tall pea plant is dominant. That last sentence is a major key, and when you see it, you should start there. What I mean is assign a letter to the dominant trait. So here I will assign the letter capital T to equal tall and to represent dominant. Next, let's write down our possible genotypes. Capital T, capital T, capital T, and little t, little t. The given information tells us both the plants are heterozygous and tall. So the genotype we're looking for must be heterozygous and it must have the dominant allele. Big T is homozygous, big T is heterozygous, and little t is homozygous. The only genotype that fits our description is big T, little t. It is heterozygous and it codes for a tall plant. So we're going to take this genotype and put it in our Punnett square. Big T, little t represents both of our parents in this particular problem. So next, let's fill in our boxes with possible offspring. The question is asking for the likely phenotypes. Phenotypes is referring to the observable traits like tall or short. 3 of these squares will yield a phenotype of tall because they all have a big T. That one left over, little t, will yield a phenotype of short plant because it is 2 little t's. The answer given by ATI was this. Heterozygous crosses results in a 3:1 phenotype ratio. In other words, we have 3 tall plants to 1 short plant. If we cross big T, little t genotype with another big T, little t genotype plant, we're going to get a 3:1 phenotype ratio. This next question is also from the ATI TEAS study guide. It states, pea plant seeds are either yellow or green. Green seeds are dominant to yellow seeds. 2 pea plants that are heterozygous for seed color are crossed. What percent of their offspring will have green seeds? So the first thing you want to examine in the given information is what is given as dominant. Here it says green seeds are dominant. So we'll first assign a capital letter to represent green seeds. I'm using the letter S for seeds. Big S will represent green seeds, and by default, little s will represent yellow seeds. Let's look at the possible genotype variations. We can have big S, which is homozygous and will yield a green seed. We can have big S, which is heterozygous and will yield a green seed. And we can have little s, which is homozygous and it will yield a yellow seed. The given information says we have 2 pea plants that are heterozygous for seed color. That's what we're working with to find our genotype for both parents. It looks like only one genotype fulfills that description. Big S, little s is heterozygous. It doesn't tell us the color it yields, but it doesn't matter in this particular problem. So we're going to fill in this Punnett square appropriately. I'll give you some time to do that. Okay, so the question is asking what percent of their offspring will have green seeds. If the offspring is to have green seeds, a big S must be present. So these 3 out of 4 will come out with green seeds. The answer given by ATI was 75% will be green. Next, we're going to try a bit of a harder question. This problem is involving 2 traits, therefore we need to upgrade to a dihybrid square. Given the following information, how many seeds will be green and wrinkled? So color is one trait and the shape is another trait. Okay, so given information is right here. From this, we can see that round shape is dominant and yellow color is dominant. We're also given the genotype for each parent. The X in the middle just means that they're being crossed. It's a bit hard to tell big Y from little y here, so I'm just going to highlight big Y for the time being. The very first thing we're going to do is FOIL. FOIL stands for first, outer, inner, last, just like algebra. So first we get big R, big Y. Outer we get big R, little y. Inner we get little r, big Y. And last we get little r, little y. Okay, so I'm going to let you try to FOIL the next one on your own. Awesome. So now we're going to use these genotype combinations and put them into our Punnett square. These are all for the first plant parent, and we will put them up here. And these are all for the second plant, and we will put them down here. By the way, it doesn't matter where you put parent 1 or parent 2. When you cross them, it's all going to be the same. So let's fill in a couple of these together. First, we have big R, big R, big Y, big Y. Next, we have big R, big R, big Y, little y. Next, we have big R, little r, big Y, big Y. And I'm just going to go ahead and give you a few minutes so that you can try and fill out the rest. Take your time because this part is really crucial to getting the correct answer. Okay, so I'm going to make some room here and push this stuff to the side. And I just want to remind you that R represents the shape of the seed. So if there's a big R present, it will give you a round seed. And if there is no big R present, then it's going to give you a wrinkled seed. Y represents trait number 2, the color of the seed. So if there's a big Y present, then the seed will be yellow. If there is no big Y present, the seed will be green. Okay, so the question is asking how many will be green and wrinkled. These 4 that I circled will be wrinkled, and these 4 will be green. Only one of these will be both wrinkled and green. So your answer is 1. Let's do a bonus question. How many of these seeds will be yellow and round? I'll give you some time to think about it. These 12 will be yellow, and these 12 will be round. So how many are both yellow and round? 9. Awesome. 9 will be both yellow and round. Okay, so just remember when you are presented with a question involving two traits, you need to use a dihybrid square with 16 boxes, and remember to FOIL first. Okay, so we're done with the fun stuff. We know how to fill in a Punnett square and all. There are a few facts you need to know as well as exceptions you need to know. So let's go ahead and review those really quick. Gregor Mendel is the father of genetics, and he studied genetics using pea plants. He found how genes are transferred from parents to their offspring. So you must be familiar with Mendel's law of heredity. There are three in total. The first is law of dominance. It states that dominant alleles mask recessive alleles, thus making the dominant alleles express. So you should understand this first law fairly well because this is exactly what we've been going through in every single practice problem. The second is law of segregation. Alleles are separated in a way that each gamete carries only one allele for each gene. This one is a bit hard to visualize, but the concept is super simple once you're able to get it, once you're able to visualize it. So if you look back at our previous problem, notice when we broke down the genotypes for the parents, in other words, we FOILed them, we got these gametes that each had either a small r or a big R, but not both. And they each had a small y or a big Y, but not both. This is what the law of segregation is. It means you won't have both alleles for one gene in one gamete. Instead, the two alleles split into two different gametes. Okay, so the third law is law of independent assortment. Alleles do not influence each other when they segregate into gametes, and thus each combination of different alleles is equally likely. Alrighty, so let's lastly go over a few exceptions to Mendel's law. These are also referred to as non-Mendelian traits. First, incomplete dominance. This is when the dominant allele is not fully expressed. In this situation, there is a blend. For example, in Mendel's law, if we have a dominant allele and a recessive allele together, dominant will automatically mask the recessive, right? And the dominant trait will completely show. So here, the dominant trait is blue. But in incomplete dominance, the dominant is only partially expressed, which means there is a blending of both the dominant color and the recessive. So for incomplete dominance, think of a blend. In codominance, both alleles are fully expressed. Emphasize on fully. So for example, we have a yellowfish and a bluefish. We end with a fish that has both fully expressed alleles, both fully expressed colors. The fish has both yellow and blue. It's not a blend. So if this was incomplete dominance, then the offspring would be a mix or blend of whatever blue and yellow make, right? Okay, so lastly, we have polygenic inheritance. This is when many genes influence one trait. For example, height is influenced by many genes. So here, AA is one gene, BB is another gene, CC is another gene, and DD is another gene. They all work together to create height. The scientific method is a systematic way to find the answers to questions or problems of the natural world. It's used to explain all of the wonders of the world and also used to question the wonders of the world. You may recall that the scientific method is performed in steps. The first step is always observation. We observe something of the natural world and we form a question or a hypothesis about it. Our second step is the hypothesis. The hypothesis is an informed, logical, plausible explanation for what we observed. One important thing about the hypothesis is it must be testable. So you must be able to construct an experiment to prove that the hypothesis is correct or not. Our third step is experiment. This is where we test our hypothesis. In our experiment, we collect measurable data that we use to analyze the result and come to a conclusion. The one most important thing that you have to remember about this step, the experiment, is that you're going to have an independent variable and a dependent variable. So let me just give you a really quick example of an experiment so you can identify the independent variable, dependent variable, and also the constant variable. Okay, so let's just say, for example, here we are conducting an experiment on a few plants. We have 3 plants here, and let's just say we want to know if feeding them a different type of liquid will change the results of the way they grow. The independent variable is the one thing that you can change in an experiment. It's the one thing that we will manipulate. And in this case, it's the type of liquid we're using on each plant. This independent variable is going to be manipulated or changed in each plant. Now, the dependent variable is the result of the independent variable. So in this case, it's what results will the plant have as an effect from the liquid that we gave them. So if we gave the first plant Coca-Cola and the plant withered up and died, then withering would be the dependent variable. That is a result of the independent variable. If the next plant we gave orange juice, and let's just say its leaves got more green and bright, then our dependent variable would be the leaves becoming bright green. This is a result of the orange juice. And let's just say that the third plant we gave water to, and this plant in particular grew very, very tall. Now, the independent variable is the water, and the dependent variable is the growth, the height of the plant. The plant grew very high. Now, everything else in the experiment must be what we call controlled. Everything must remain constant and unchanged. Therefore, we know that what is causing the dependent variable or the outcome or the data, the results, is in fact the independent variable, the change in the type of liquid that we're feeding each plant. Some examples of a constant variable in this particular example could be the amount of liquid that we give each plant. So you see that's different than the type of liquid we're giving each plant, right? So the amount of liquid has to be constant. The amount of sunlight we give each plant can be a constant. The type of soil we're giving each plant can be a constant. And let's say how big each pot that we put each plant in can be a constant. So everything else, all other variables must be constant except for the independent variable, which is being manipulated. Alrighty, so as far as the experiment portion of the scientific method goes, I personally feel it's the most important. You must know the difference between independent variable and dependent variable. And if somebody gives you a question, you must be able to, if someone gives you an experiment, you must be able to point out the independent and dependent variables in the experiment. The final step of the scientific method is the conclusion. In the conclusion, there are 3 important things that you must include. The first is the summary of the overall experiment. The second is the results of the experiment. And the third is whether you accept or reject the hypothesis. Next, we're going to jump into the biological hierarchy. This is just a systematic way of putting every living thing into levels of complexity. So we begin first with the atom. The atom is what makes up all matter. It also makes up molecules. Molecules are any 2 or more atoms put together by a chemical bond. Molecules are what make up cells. Cells are the basic building blocks of all living things. Cells are what make up our tissues, and our tissue is a group of cells that have similar structure and that function together as a unit. Tissues are what make up our organs, and an organ is a collection of different types of tissue that structurally form a functional unit specialized to perform a specific function. Our organs create our organ systems. One example of an organ system is the cardiovascular system, and its main organ is the heart, which helps pump blood, but it also includes the accessories to the heart that help the heart pump blood and take it to where it needs to go. So it includes all of the blood vessels that is very crucial, that work hand in hand with the heart to spread blood throughout the body. Now, our organ systems work together to create an organism. An organism is an individual, animal, plant, or single-celled life form. So any living thing, any singular living thing is an organism. And any organism is going to have all of the things we just discussed included in it. So it's going to have organ systems, organs, tissue, cells, molecules, atoms, etc. After organism, we have population. When you have a group of organisms of the same species grouped together, we call this a population. So for example, we have a group of humans, the human species, and they're grouped together. So this is one population. In this other photo, I have a group of bats hanging out together, and they are of the same species. And they are multiple organisms hanging out together. So we call this a population. Now, populations build communities. And a community is a collection of different types of populations of species living together. So a lot of people get population and community mixed up. So the major difference is that population is referring to one particular species living together, and community is referring to different populations of different species living together. So there is a population of these bats, and they actually live in this community in this cave in New Zealand. The reason why this cave is a community is because there are different types of species living in it. There is the population of bats, but there's also this population of cave fish that you see this guy holding. And there's also this population of this really gnarly fungus that is glow in the dark that you can see here. We have these three different types of population, these three population of three different species living together. That makes this a community. So after we have community, we have an ecosystem. An ecosystem is all of the communities in a particular area, but it also includes the physical environment in which they live in. Ecosystem basically includes the community and population and all the organisms, but added to it is the physical environment, like the trees, the ocean, the grasslands, the mountains, everything in which they live in that is part of Earth, that is an ecosystem. Next, we have a biome. A biome is a really, really large area, environmental area that has very unique characteristics to that area. It shares very similar types of temperature, soil type, maybe water type, amount of light, similar type of bacteria, flora, fungi, fauna, things like this, similar types of species like animals that occupy this area. So our biomes are a collection of all the different ecosystems, but they share very similar climate conditions or environmental conditions in general. So here I have a picture of some of our most common biomes. We have grassland, savanna, desert, marine, freshwater, tundra. All of these have very distinct, let's just say, temperature ranges, soil type, species, and the picture kind of makes it very easy to see the drastic differences. So moving on, lastly, we have our biosphere. The biosphere includes all of the biomes within it, and it includes all the places on Earth where life exists. And this, my friend, completes the biological hierarchy. Alrighty guys, moving on to the pH scale. Although pH scale is super simple and there's not much you need to memorize about it. It's still very important in biology, and it's one of those high yield questions that you might get on your TEAS exam. So if you know the basics of the pH scale, you can go ahead and skip ahead, but if you need a refresher, stick around. This will only take a minute. First thing first, the pH scale is the measurement of hydrogen ion concentration in a water-based substance. So your acids are going to have a high concentration of hydrogen ions, but your bases are going to have low concentrations of hydrogen ions. So you have your acidic on one side and your alkaline or base on the other side. And you have your neutral smack in the middle. Your neutral is number 7 and only number 7. Every other number belongs to either an alkaline or an acidic. So our acidic solutions are going to be numbered 1 to 6, and our alkaline solutions are going to be numbered 8 to 14. So here are some very common solutions that you will find on our pH scale. Remember that number 7 is your neutral. The further you go to your right, closer to 14, the more alkaline or basic a substance gets. And the further downwards you go to 0, the more acidic solution gets. Alrighty everyone, so we're going to move on to macromolecules, or what it's also called, the organic molecules. Now, there are 4 macromolecules: proteins, carbohydrates, lipids, and nucleic acids. As far as the ATI TEAS exam goes, there's really only 3 things you must know about each of these. 1 is what they're composed of or made of. 2 is the structure of the molecule, and 3 is their functions in our body. The 4 macronutrients that we're going to be studying are the carbohydrates, lipids, proteins, and nucleic acids. Macronutrients are nutrients required in large amounts that provide energy needed to maintain bodily functions and carry out the activities of daily life. Macromolecules are associated with this process called hydrolysis. Hydrolysis means that when the molecules enter water, when they're immersed in water, they can be easily broken down into smaller molecules. So essentially, we have these macromolecules like fats, carbohydrates, and proteins. And when they enter our digestive system, the water breaks them down, also with the help of enzymes, breaks them down so that those nutrients or macromolecules are easily absorbed into the bloodstream. So just remember that word hydrolysis. The true definition of it is the chemical breakdown of a compound due to reaction with water. So just remember that in hydrolysis process, compounds are immersed in water and enzymes help break down these compounds into smaller substances. So let's first start with what these macromolecules are made out of. They're made out of something called monomers, and monomers are just building blocks of smaller molecules that build big molecules. Carbohydrates are made out of a monomer called monosaccharides. Lipids are made out of fatty acids and glycerol. Proteins are made out of amino acids. And nucleic acids are made out of nucleotides. All of these are monomers that help build our macromolecules. So how are these 4 macromolecules used in our body? Starting with the carbohydrates. Carbohydrates are used as a fuel for our body. Your body breaks down carbohydrates into glucose, and glucose is the main source of energy for your body's cells, tissue, and organs. There are 3 types of carbohydrates. There's your monosaccharides, disaccharides, and polysaccharides. Remember that mono is referring to one or singular. So monosaccharides are your singular simple sugar. Di means 2. So disaccharides are 2 monosaccharides that are bonded together. Poly means many. So polysaccharide is referring to a long chain of multiple or many monosaccharides. Common monosaccharides are glucose and fructose. Common disaccharides are sucrose and lactose. And common polysaccharides are starch, cellulose, and glycogen. So next we have our lipids, and remember that the monomer of the lipids is fatty acids and glycerol. These are the building blocks of lipids. It's what make up the lipids. So we're going to go into a lot more detail with the lipids because there's a lot to know here. So hang in there, guys. If you're not already bored, you might just get bored here. So just hang in there. So first and foremost, when we talk about lipids, we're talking about fats. And there are three types of lipids. There are your triglycerides, your phospholipids, and your steroids. Triglycerides include your animal fats and your oils. There are significant differences between these two. Animal fats are normally solid at room temperature, and oils like vegetable oil, avocado oil, olive oil, these are liquid at room temperature. And the reason why animal fats become solid and oils are kept as liquid has to do with their molecular structure. So your animal fats are what we call saturated fats, and your oils are what we call unsaturated fats. On a molecular level, this is what it's going to look like. Saturated fats is going to have generally a carbon-hydrogen atom chain, and all of your carbon atoms are going to be connected or bonded by a single bond. So single bond is represented by this single line. So that's the takeaway here, is that your saturated fats, your carbons are going to be bonded by a single bond. There's going to be single lines between them. Now, your unsaturated fats, they look a little bit different because they have at least one double bond between their carbons. So it might look a little bit like this. You have carbons with single lines, but maybe you have one or two double bonded carbons linked to each other. This is going to make it a unsaturated fat. And in fact, these double bonds is what changes the melting point of the substance. So this is what makes these oils liquid at room temperature, basically. So this is generally what you need to know. Triglycerides, again, you have your animal fats and your oils. Your animal fats are saturated fats, meaning molecularly the carbon to carbon bonding is going to be singular. And then your oils are unsaturated fats, meaning on a molecular level, your carbon to carbon, you're going to have at least one double bond. And also remember that saturated fats are solid at room temperature, like your animal meats, and your unsaturated fats are liquid at room temperature. Let's go ahead and move on to our second type of lipid, the phospholipid. So the major thing about a phospholipid is that it has two sides to it. It has this head that is made out of glycerol, and remember that glycerol is one of the monomers that help build and construct the lipid. And attached to this glycerol is a phosphate group. Now, this phosphate group, molecularly, it would look something like this, but what's really important about this phosphate group is that it makes this part of the phospholipid, it makes it hydrophilic, meaning hydro meaning water, philic meaning loving. So this part of the phospholipid is water loving. And the other side of the phospholipid are these two little legs that come out here, and these are made out of fatty acids. Now these fatty acids are, these are called the tails. My bad, not the legs, the tails. So these tails are made out of fatty acids, and they are actually hydrophobic, meaning they hate the water. So to sum that up, phospholipids have a hydrophobic side and a hydrophilic side. Your phospholipids form a bilayer, and this is called or referred to as a lipid bilayer or a phospholipid bilayer. Now, your outer part of the layer is going to be the head or the hydrophilic side of this lipid. This is the water-loving side. And on the inside of the layer is going to be your hydrophobic or your water-hating side. It's going to be safely on the interior. These phospholipid bilayers are super important because they make up our cell membranes. They are a protective layer of our cell membranes, specifically because they have the hydrophobic and hydrophilic side. So for most organisms and most viruses, this is going to make up a major part of your cells. So the final lipid that we're going to learn about are our steroids. A steroid is any compound with a specific 4-ring structure. Now, steroids apply to various types of chemicals. One important steroid is cholesterol. Cholesterol is important because it is a precursor to the synthesis of other steroids such as testosterone, estrogen, vitamin D, and cortisol. Another common steroid is aldosterone. Aldosterone maintains fluid balance in your body by promoting reabsorption of excess sodium and excreting potassium. The last steroid I want to touch on is cortisol. Cortisol is important because it has anti-inflammatory effects on the body. Related to cortisone is a classification of drugs, man-made drugs, called corticosteroids. These are a type of anti-inflammatory drugs, just like cortisone, anti-inflammatory. These are given to patients for treatment like rheumatoid, arthritis, lupus, vasculitis, things like this that have that the cause is too much inflammation in the body. So in my opinion, as far as the ATI TEAS exam goes, there's not too much you need to know about steroids. The most important thing is that what is unique to them is that 4-ring structure. So if you see the 4-ring structure, you're going to need to identify it as a steroid, but also know some of the high yield types of steroids. From my research, through various books that have review questions, I've seen aldosterone, cortisol, vitamin D, testosterone, and estrogen. And I think testosterone, estrogen, and what's the other one? Progesterone. Progesterone is also one of those high yield steroids. First off, remember that monomers refers to the building blocks of macromolecules. Monomers are basically smaller molecules that make up bigger molecules. So the specific monomer of a protein are called amino acids. And these amino acids are referred to as polypeptides. Proteins have a different structure depending on how many proteins are present. The primary structure is a simple chain of amino acids, and this is called your amino acid sequence, also referred to as your polypeptide chain. There are 21 common amino acids that the body needs to function and to grow. 9 of these are called your essential amino acids. Now, these 9 essential amino acids cannot be synthesized by the body, meaning they cannot be made by the body. In other words, you're going to need to get them through your diet, through foods. Let's go over a few types of proteins. The first one is structural protein. This is going to make things hard and rigid. A good example of a structural protein is keratin. Keratin is a type of protein that forms your hair and your nails. It's what makes these things hard and rigid. Next, we have our enzymes. Enzymes catalyze chemical reactions. In other words, they accelerate the process of chemical reactions. Without these enzymes, some chemical reactions would never occur. The third type of protein are some hormones. So some hormones are made out of proteins or peptides, and others are made out of steroids. And remember, a side note, the steroid is a lipid. A few important hormones that are made out of proteins are insulin, glucagon, and pituitary hormones. Next, we have a receptor. A receptor is a protein molecule that's usually found embedded within the plasma membrane surface of a cell. This receives chemical signals from outside of the cell, and when such signals bind to the receptor, they cause some sort of action or some sort of cellular or tissue or biological response. Next, we have antibodies. Antibodies are also known as immunoglobulins. They bind to antigens and they target them for destruction. So they work in our immune system to keep us safe from foreign bodies. Lastly, we have motor proteins. They convert chemical energy into mechanical work by the hydrolysis of ATP. Basically, they generate the forces that cause our muscles to contract. Alrighty guys, so this concludes what we need to know about proteins. So we are going to move on to our last macromolecule, which are the nucleic acids. Our nucleic acids include DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. Our nucleic acids are made out of the monomer called nucleotides. Remember, monomers are the building blocks of our macromolecules. Nucleotides consist of 3 portions. They have a phosphate, a sugar, and a nitrogenous base. Nucleotides make up both DNA and RNA. The nitrogenous bases in DNA are adenine, thymine, guanine, and cytosine. Adenine and thymine always pair together, and guanine and cytosine always pair together. Now let's take a look at the nitrogenous bases in RNA. We have adenine and uracil, guanine and cytosine. So adenine and uracil pair together, and guanine and cytosine pair together. What are cells? Cells are the basic building block of all living things, and cells also have the important job of carrying around DNA for all living organisms. So because cells make up basically all living things, the cell theory is so significant because it states the inevitable rules of the cells themselves. So it comes in 3 parts. The first is that all organisms are made of cells. The second is that cells are the basic unit of all living things. And the third is that cells are made from pre-existing cells. Keep in mind that this definition of modern cell theory is super simplified. It's basically a general understanding of what it is. So if you look through a textbook or another resource, you might see a very extended and detailed version of the modern cell theory. However, these 3 sentences basically just break it down. So now what kind of cells are there? Well, all cells of living things fall into one of these 2 categories. Either the cells are prokaryotic cells or eukaryotic cells. So when we say prokaryotic, we're referring to the cell itself or the type of cell itself. When we say prokaryote, this is referring to the organism that is made up of prokaryotic cells. When we say eukaryotic, this refers to the cell itself. And when we say eukaryote, this is referring to the organism that is made out of eukaryotic cells. So your prokaryotic and eukaryotic cells actually have a lot more in common than they do differences. Nevertheless, they do have one major difference. So prokaryotic cells lack a nucleus and a few other organelles. However, they lack a nucleus, and this is the big takeaway. Your prokaryotic cells include bacteria, archaea, and cyanobacteria. So in contrast to eukaryotes or eukaryotic cells, they have a nucleus. This is the major difference between the eukaryotes and the prokaryotes. So eukaryotes have a nucleus, and these include your plants, your animals, and your fungi. So we're going to focus in on our eukaryotes because mainly when we talk about cells, we're talking about eukaryotic cells. Like when we're talking about the human cells, they're all eukaryotic cells. Correction. So all human cells are eukaryotic cells. However, there are colonies of bacteria that is normal in our stomach and our digestive tract, and these are known as like probiotics. So these bacterial cells are prokaryotic because remember, prokaryotic includes bacteria. So let's talk about the differences between plant cells and animal cells. So plant cells have a cell wall that surrounds their cell membrane. Both plant and animal cells have a cell membrane that surrounds their cell and protects it. However, plants have an extra durable cell wall that is made out of cellulose, and this just adds extra strength and extra protection to the plant cell. So another major difference between plants and animals is the way that they get their food or energy. Plants are what you call autotrophs, and animals are what you call heterotrophs. So autotrophs make their own food. Plants and algae are great examples of autotrophs. They basically take the energy of the sun and they transform it into glucose, which is the food source and energy source for these plants. And this process is called photosynthesis. So humans and animals, on the other hand, we cannot do photosynthesis. So we are called consumers, or heterotrophs are called consumers, because they must consume something else in order to get their food and their energy. So these are the major differences between plants and animal cells. And remember that both plants and animal cells are eukaryotic cells. So now let's talk about the similarities between the two. Both plants and animal cells have what we call a membrane-bound organelles. So what this basically means is that each organelle within the cell has its own protective layer surrounding it. So they have something called a phospholipid bilayer, which we'll go ahead and take a look at a little bit later. But just keep in mind that this protective phospholipid bilayer is used to regulate what comes in and out of each organelle. There is one exception to these organelles. The ribosomes are the one that do not have a phospholipid bilayer, so ribosomes are not membrane-bound organelles. Okay, so secondly, plants and animals, they both have the main organelles like your mitochondria, nucleus, endoplasmic reticulum, the Golgi apparatus, and peroxisomes, and they both also have the cytoskeleton. So notice that I left out the lysosomes, and that's because yes, both animals and plants do have lysosomes. However, it is far less likely to find them in the plant cell. So now we know what a cell is, what type of cells there are, the differences between eukaryotic cells and prokaryotic cells, and the differences between plant and animal cells. So now let's go ahead and look at the structures of a cell, in other words, the organelles in a cell, starting off with the cytoplasm. Your cytoplasm is everything that is within the cell walls. So all of the organelles within the cell walls and also the fluid that surrounds these organelles, this is all part of the cytoplasm. There's also something called the cytosol. Cytosol is all of that matrix jelly-like liquid substance that surrounds all of the organelles. So remember, cytoplasm and cytosol. Your cytosol is within the cytoplasm or is a part of the cytoplasm. So next we have our cell membrane or our cell wall, and it's composed of this phospholipid bilayer. Bi meaning two, so it's a two layers or double layers of phospholipid. In our phospholipid bilayer, we have a polar side and a non-polar side. The head are polar, polar meaning that they are able to interact with water. They are water-loving. So water-loving is called hydrophilic. So this head interacts with water, and it's important that it's on the outer layer of this phospholipid bilayer because we have water on the inside of the cell and water on the outside of the cell. So it stays on the outer layer of this phospholipid bilayer so that it can easily interact with the molecules within the liquid or within the water. So the head is polar and hydrophilic, while the tails that are hiding on the inner side, these are non-polar. They do not like the water and they do not react well in the water. So these are called hydrophobic. So the cell wall is a semi-permeable membrane that acts as a barrier. It carefully selects what can come inside the cell and what can leave the cell. There are some really important molecules that hang out around this phospholipid bilayer, and one of them is cholesterol. Cholesterol regulates the fluidity between the phospholipids, and fluidity is basically referring to the space between these phospholipids. The more space there is between them, the better chance molecules have of getting into and out of the cell. Now, this can be harmful if the space or the gaps are way too large between these phospholipids because it will be harder for the cell membrane to control what comes in and out of the cell. Now, in contrast, if there is very little space between these phospholipids, then it will be harder for bigger molecules to get in and out of the cell membrane. So having too much space or too little space is both harmful to the cell. Temperature has a lot to do with this. So the lower the temperature is, the more compact and rigid these phosphomolecules are going to be. Now, how this works is the cholesterol is going to come in and act as spacers and space out these phospholipids so that it is easier for molecules to come in and out. Now, the opposite happens when the temperature is too high. When it's too high, then these phospholipids become way too spaced out and they need cholesterol to come in and kind of squeeze them back together. So basically, cholesterol controls fluidity. When it is too cold or the temperatures are too low, the phospholipids are going to try and become too compact and close together, and the cholesterol is going to come in and act as a spacer. When the temperature is too hot or too high, then cholesterol is going to come in and squeeze these phospholipids together so that there's not too many gaps between them. So your second important molecule that hangs out here in the phospholipid bilayer are your proteins. Now, there are two very important proteins, your peripheral proteins and your integral proteins. Peripheral proteins hang out in the peripheral. They don't go into that inner hydrophobic area. They hang out on the outside. And these help in transporting materials in and out of the cell. Now, your integral proteins help with signaling and communicating, as well as moving molecules through the plasma membrane. So just remember here, guys, that we're talking about your cell membrane when we talk about the phospholipid bilayer. And also remember that the most important molecules that are embedded within this phospholipid bilayer are your proteins and your cholesterol. So next we have our nucleus, and it has a few important components to it. First, we have our nuclear envelope that surrounds our nucleus. The nuclear envelope is two layers. The outer layer has ribosomes and the inner layer has lamins. Lamins is a type of protein that has many important roles, but one of the most important is it plays in cell division. Now, the outer layer that has ribosomes, well, we're going to go over ribosomes later, but one thing that's very important is that inside of the nucleus, mRNA is made, and then the mRNA travels to a ribosome, and the ribosome carries it to the rough ER. So a part of your nuclear envelope are these pores, and these pores are made out of proteins. And basically the pores help regulate what comes in the nucleus and what goes out of the nucleus. So next, within the nucleus, we have this thing called a nucleolus, and it's a tiny little sphere that is the site of our RNA synthesis. In other words, our RNA, ribosomal RNA, is made here. And remember that our RNA has such an important role in protein synthesis. Remember, so we'll go over that when we start talking about the ribosomes. But remember, our RNA synthesis is right here in the nucleus. So lastly, we have something called chromatin. Chromatin actually looks like this. It's more like squiggly and loose little strings that are inside of the nucleus, and they're made of DNA, RNA, and proteins, specifically histone proteins. Chromatin is the material that makes up chromosomes. So chromosomes are made out of chromatin. So that's it for the nucleus. So just remember, it has the nuclear envelope, the nuclear pores made out of proteins. You have your lamins inside of the inner layer of the envelope and your ribosomes on the outer layer. You have your nucleolus, which is the site of our RNA synthesis, and then you have your chromatin, and your chromatin is what makes your chromosomes. So let's go ahead and move on to the rough endoplasmic reticulum. So the rough endoplasmic reticulum, or rough ER for short, is covered with ribosomes, and this is its main characteristics. One important thing that happens within the ribosome specifically is translation. Remember, transcription and translation. Transcription is when DNA makes mRNA, and translation is when mRNA translates or converts into amino acid chains, which is a protein. So translation is part of protein synthesis, and this happens specifically in the ribosomes in the rough endoplasmic reticulum. So once these proteins are made, the rough ER helps fold the protein, and it also packages the protein into a vesicle and pushes it out towards the Golgi apparatus. And remember, a vesicle is basically like a packaging box that keeps the molecules safe. The rough ER also participates in a process called glycosylation. Glycosylation is the activation of a protein. So basically it takes these enzymes that are within the rough ER and it adds some sugar residue to the protein, and this activates it. So rough ER, it is the site for protein synthesis, specifically in the ribosomes. It folds the protein, it packages the protein to be pushed out within a vesicle, and it also is the site of glycosylation. Next, we have the smooth endoplasmic reticulum. It's smooth because it has no ribosomes, but it does have a ton of enzymes, and these enzymes help make or create lipids like fatty acids, phospholipids, and cholesterol. So we can say that the smooth ER is a site for lipid synthesis. It also makes an enzyme that is super important and found in the liver. It's called CY450. It's found in the liver because it aids in detoxification. Lastly, the smooth endoplasmic reticulum is able to store calcium. So it basically has these little pumps, and calcium is stored here. Whenever calcium is needed in the blood, then these pumps just push out a little bit of calcium at a time for us to use. Next, we have our ribosomes. Remember, ribosomes are found on the rough ER, and that's what makes them rough. So these ribosomes are called the membrane-bound ribosomes. They're also found floating around freely in the cytosol. So these are called free ribosomes or cytosolic ribosomes. So ribosomes actually look a little more like this. They're not just like one flat circle. They have two parts. They have a large top and they have a smaller bottom. Most distinctly, the ribosomes are known to be the site of protein synthesis. And I'm not going to go into too much detail with this because I went into detail in my last video about protein synthesis. So if you're a little bit lost, go back to my last video and check that out. Basically, what happens here is translation. And remember, translation is the second part of protein synthesis. It's when mRNA goes into the ribosomes and it is translated into amino acids. And remember that amino acids are what make your proteins. So translation here happens in the ribosomes, and it is the site of protein synthesis. The Golgi apparatus, first off, it receives vesicles from the smooth ER and the rough ER. And remember, what's coming out of the smooth ER is the lipids, and what's coming out of the rough ER are the proteins. So proteins and lipids are being delivered to the Golgi apparatus. Here, the Golgi apparatus modifies these proteins and lipids using processes like glycosylation and phosphorylation. So once it does this, it packs them up into vesicles, both the protein and the lipids, and it sends them off where they need to go. There are a few parts to the Golgi apparatus that are important to remember, and one is the cis phase. The cis phase is the part of the Golgi where molecules come inwards into the Golgi. So molecules like proteins and lipids are going to enter through the cis phase. Once they're inside of the Golgi, they go through things like glycosylation, phosphorylation, which are what we call modifications, and then they get packaged into vesicles. Once they're packaged in the vesicles, these molecules leave the Golgi apparatus through the trans phase. So just remember, the cis phase is molecules entering the Golgi, and the trans phase is molecules leaving the Golgi. Next, we have our lysosome. Our lysosome is kind of like a cleanup crew. It breaks down macromolecules. Remember, our macromolecules include protein, lipids, nucleic acids, and carbohydrates. Lysosomes have special enzymes that can break down each of these specifically. So, for example, protease is going to break down our protein. Lipase is going to break down our lipids. Nuclease is going to break down our nucleic acids, and glycosidase is going to break down our carbohydrates. The lysosome is also able to break down old, worn out organelles as well as cells that are no longer able to function. Basically, they're also old and worn out. Autophagy is when the lysosome takes the old, worn out organelles and breaks them down. And autolysis is when the lysosome takes the old, worn out cells and breaks them down. So autophagy and autolysis, this is something that the lysosomes also do. Next, we have the peroxisomes. Peroxisomes help defend us from free radicals. It does this because it has enzymes, and some of the most important enzymes that it has is catalase and oxidase. These metabolic enzymes help convert or break down these free radicals and turn them into not so dangerous molecules. So in this process of breaking down these free radicals, peroxisomes excrete hydrogen peroxide, so hence the name peroxisome. Next, we have the mitochondria. The mitochondria has two important components to it, the outer membrane and the inner membrane. So our outer membrane has an increase in permeability. Permeability means the ability for things to come in and out of it or through it, right? So an increase in membrane permeability means that it's going to be easier for molecules to come in and out of there. Secondly, we have the inner membrane where there is little or less permeability. So it's going to be harder for molecules to come in and out of the inner membrane. So outer membrane, increase permeability, inner membrane, less permeability. Okay, so there are a couple functions to the mitochondria. The mitochondria, one really cool fact is that it has its own mitochondrial DNA, and this comes from the mother. And secondly, it has a ton of metabolic reactions. But we are not going to go over all of these metabolic reactions because this would take forever, but just know that it has very important metabolic reactions. One other very important thing is that it is the site of ATP synthesis, and that's why we call the mitochondria the powerhouse. It generates most of the chemical energy needed to power the cell's biochemical reaction. So it makes ATP, and this is what gives the whole cell energy to perform or to function. Okay guys, so lastly we have our cytoskeleton. The cytoskeleton is kind of like the glue that keeps everything together inside of the cell. We have three important types of cytoskeleton. One is the microfilaments or the actin, secondly is the intermediate filaments, and third is the microtubules. So first let's talk about microfilaments. Microfilaments or actin help in muscle contraction as well as they control the shape of cells. So this is how they do it. If you recall, during muscle contraction and relaxation, actin works with myosin to contract and relax the muscle. So in the same way that actin helps this contraction, it also helps a single cell kind of contract in a way. So let's just say if a cell needs to be split into two or split apart, actin is going to kind of go around it and constrict it, kind of like a waist trainer, basically. It's going to constrict it so tight and squeeze it so tight that it's going to make the cell split into two. Also, let's just say a cell wants to squeeze into a tight space. Actin is going to come and surround a part of the cell, kind of like a little waist trainer, and it's going to squeeze it and shape it into a way that it can fit through the small space. So basically, actin helps in muscle contraction, but it also forms a ring around cells so that it can shape it to go through small spaces, but also it can help split the cell in half. Next, we have the intermediate filaments. These are stiff and rigid type of fibers, and they are going to help anchor your cell to your cell, as well as your organelles inside of the cell to the cell, as well as the cell to the extracellular matrix. Remember, extracellular matrix is all of that fluid or jelly that surrounds each cell. So basically, intermediate filaments attach all three of these things together so that they're not just floating around everywhere getting lost. Lastly, we have our microtubules. So these microtubules have an important protein called motor proteins within them. These motor proteins are going to help with movement. So they help with intercellular transport, cell division, and cell extensions. This is how. First off, the microtubules are able to bind themselves and attach themselves to molecules. Once they bind themselves, they're able to transport the molecules from one place to another within the cell. Secondly, they help in cell division, specifically the metaphase, because the microtubules attach themselves to the chromosome, sort of like Legos. They are able to attach, but also make a split and separate these chromosomes. And lastly, these microtubules form some cellular extensions. So cellular extensions are things like cilia and flagella, things that need to, beat back and forth really rapidly. They're going to need these motor proteins within the microtubules. Alrighty, everyone, that's it for this video. Thanks for sticking around. I hope you learned something new, and until next time.